When you look at your reflection in still water, you're witnessing one of nature's most fundamental patterns. A pattern so ancient, so deeply woven into the fabric of life itself, that it shapes everything from the smallest flower to the largest whale. Welcome to Bedtime Biology, where science meets serenity. Each night, we explore the quiet wonders of the natural world, calming your mind and sparking your curiosity. Tonight, we're embarking on a gentle journey through one of nature's most elegant principles, symmetry. It's everywhere around us, yet often invisible until we pause to notice. We'll discover how a butterfly's wings mirror each other perfectly, how a starfish defies the rules we think we know, and how a snowflake captures mathematical perfection in frozen water. We'll explore flowers that use symmetry to seduce pollinators, animals that abandoned symmetry entirely, and the invisible forces that turn a simple pattern into the foundation of consciousness itself. The story of symmetry is the story of life's search for balance, efficiency, and beauty. It's written in the spiral of a nautilus shell, the petals of a daisy, and even in the structure of your own body. Some of what you'll hear tonight may change how you see the world around you. And if these calm explorations help you unwind, consider sharing them with others who might find peace in understanding the quiet logic of nature. Now, let's begin our journey into the power of symmetry, the language of patterns. Before we step into forests and oceans, let's understand what symmetry really means. In the simplest terms, symmetry is a balanced arrangement where parts on one side match parts on another. It's nature's way of organizing complexity into recognizable patterns. But this isn't just about aesthetics, though symmetry is undeniably beautiful. This is about survival, efficiency, and the very architecture of life. Imagine you're looking at a butterfly. Draw an invisible line down its center, from head to tail, and you'll notice something remarkable. The left wing mirrors the right wing almost exactly. The patterns, the colors, the shapes, they all match. This is bilateral symmetry, and it's the most common form in the animal kingdom. The word bilateral comes from Latin, meaning two sides, and that's precisely what it describes. One plane, one line, dividing the organism into two nearly identical halves. But bilateral symmetry is just one dialect in nature's language. There's also radial symmetry, where body parts are arranged around a central point, like the spokes of a wheel. Picture a sea anemone, its tentacles spreading outward in all directions from its mouth. You could slice through this creature at almost any angle through the center, and you'd get matching halves. It's the symmetry of a circle, a design that lets an organism interact with its environment equally from all sides. Then there's the rarest form, asymmetry, or the complete absence of symmetry. Sponges are perhaps the most famous example. These ancient creatures, among the simplest animals on Earth, have no recognizable pattern to their body structure. They grow in irregular, unpredictable forms, shaped more by their environment than by any internal blueprint. And yet, they've survived for over 600 million years, proving that symmetry, while powerful, isn't the only path to success. The prevalence of symmetry in nature isn't random. It's the result of hundreds of millions of years of evolution, of natural selection, favoring certain body plans over others. When a trait appears so consistently across so many different species, separated by vast stretches of evolutionary time, we know it must offer significant advantages. Symmetry helps organisms move efficiently, process information from their environment, capture food, and avoid becoming food themselves. Consider for a moment how a fish swims. Its body is bilaterally symmetrical, with fins on each side, muscles arranged in matching segments. When it wants to swim straight ahead, both sides of its body work in perfect coordination. If one side were larger or stronger than the other, the fish would veer off course, wasting energy and becoming an easy target for predators. Symmetry ensures balanced movement, and in the water, where resistance is high, efficiency can mean the difference between life and death. But symmetry offers more than just mechanical advantages. It also influences how organisms sense and process the world. Animals with bilateral symmetry typically have sensory organs, eyes, ears, antennae, positioned symmetrically on either side of their body. This allows them to detect stimuli from multiple directions simultaneously, and to judge distances accurately through triangulation. Your own depth perception works this way, your brain comparing the slightly different images from your two eyes to construct a three-dimensional understanding of space. Throughout the animal kingdom, we see that symmetry correlates strongly with complexity. The simplest animals tend to have radial symmetry or no symmetry at all. As we move up the evolutionary tree toward more complex, active, intelligent creatures, bilateral symmetry becomes the dominant pattern. This isn't coincidence. Bilateral symmetry enables something called cephalization, the concentration of nerve cells and sensory organs at one end of the body. And cephalization, as we'll explore, is the foundation of everything from simple ganglia in worms to the human brain. The study of symmetry in biology reveals deep truths about how life evolves and adapts. It shows us that nature tends towards certain solutions, certain patterns that work so well, they appear again and again across different lineages. A butterfly and a shark look nothing alike, yet both possess bilateral symmetry. A jellyfish and a daisy seem to inhabit different worlds, yet both display radial symmetry. These patterns transcend the boundaries of taxonomy, connecting organisms that haven't shared a common ancestor for hundreds of millions of years. As we journey deeper tonight, we'll see how symmetry shapes not just individual organisms, but entire ecosystems. We'll discover how the symmetry of a flower determines which insects can pollinate it, how the symmetry of a starfish hides a deeper bilateral story, and how the molecular symmetry of water creates the infinite variety of snowflakes. We'll explore how some organisms break the rules of symmetry and thrive, and how the mathematical patterns underlying symmetry connect the smallest atom to the largest galaxy. The world of symmetry is vast and intricate, a testament to nature's endless creativity, working within the constraints of physics and chemistry. It reminds us that beauty and function are not separate in nature. They emerge together, sculpted by the patient hand of evolution, refined over countless generations, until what works and what captivates the eye become one and the same. Bilateral symmetry, the mirror of motion. Let's step more deeply into the world of bilateral symmetry, the body plan that dominates the animal kingdom. When you look in a mirror, you see this principle embodied. Your left side mirrors your right side. 2 eyes, 2 ears, 2 arms, 2 legs, all arranged along a single plane of symmetry, running from head to toe. This isn't just how humans are built. It's the fundamental architecture for the vast majority of animals on Earth. Scientists estimate that roughly 99% of animal species possess bilateral symmetry. From the tiniest insects to the largest whales, from worms that burrow through soil to birds that soar through the sky, the bilateral body plan proves itself again and again as an exceptionally successful design. This pattern is so ancient that it dates back over 500 million years, to a time when the first bilaterally symmetrical animals began to dominate the seas. The formal name for this group is Bilateria, and it includes almost every animal you're likely to encounter in your daily life. Mammals, birds, reptiles, amphibians, fish, all bilaterally symmetrical. Insects, spiders, crustaceans, mollusks like snails and octopuses, all bilaterally symmetrical. Even creatures that seem at first glance to lack obvious symmetry, like certain flatworms or sea stars, evolved from bilaterally symmetrical ancestors and retain that symmetry during their development. But what makes bilateral symmetry so advantageous? The answer lies in movement. Animals with bilateral symmetry have a distinct front end, called the anterior, and a back end, called the posterior. They have a top, or dorsal, side, and a bottom, or ventral side. This creates directionality, a clear sense of forward and backward. When an organism moves, it moves in one consistent direction, with specific parts of its body leading the way. Think about how you walk. You move forward, facing the direction you're going. Your eyes look ahead, not backward. Your arms swing in coordination with your legs. Your entire body is organized around the assumption that you'll be moving in a particular direction. This seems obvious, even trivial, until you consider what life would be like without it. Now, imagine trying to navigate the world with radial symmetry, like a sea anemone. You could move in any direction, sure, but no direction would be privileged over any other. You wouldn't have a front to lead with, no natural forward. Coordinated, directed movement would be much more difficult. This is why most radially symmetrical animals are either sessile, meaning they stay in one place, or they drift with currents. The few that do move actively, like some sea stars, do so very slowly. Bilateral symmetry solves this problem elegantly. By creating distinct ends and sides, it allows for streamlined movement through a medium, whether that medium is water, air, or earth. The front end can be shaped to reduce resistance, while the back end can house propulsion systems like fins, wings, or legs. The arrangement of muscles can be symmetrical too, ensuring that the left side and right side work in perfect coordination, preventing the organism from veering off course. This streamlining is especially important in water, where even small differences in shape can have large effects on drag. Look at a fish, and you'll see bilateral symmetry expressed in its most hydrodynamic form. The body tapers at both ends. The fins are paired and positioned symmetrically, and the internal organs are arranged to maintain balance. The fish can dart left or right by adjusting the movement of its paired fins, but its default mode is straight ahead swimming, powered by the symmetrical contractions of muscles along its body. On land, bilateral symmetry enables walking, running, and jumping. Consider a horse galloping across a field. Its forelegs move in coordinated pairs, left and right, working together in precise timing. If the left legs were longer than the right legs, or if the muscles on one side were weaker, the horse would struggle to maintain balance and speed. Symmetry ensures that power is distributed evenly, that weight is balanced properly, and that the animal can change direction quickly when needed. In the air, bilateral symmetry becomes even more critical. Birds must maintain perfect balance while flying, and any asymmetry in their wings would make flight impossible. Watch a hawk soaring on thermal currents, its wings spread wide. Those wings are near perfect mirrors of each other, with the same length, the same arrangement of feathers, the same muscular control. Even tiny adjustments, spreading one wing slightly more than the other, allow the bird to turn or maintain stability in wind. But bilateral symmetry offers more than just mechanical advantages for movement. It also enables an incredibly important evolutionary innovation: cephalization, the development of a distinct head region. Because bilaterally symmetrical, animals move in a consistent direction. The front end of their body encounters new stimuli first. It makes sense then for sensory organs to be concentrated at the front, where they can detect food, predators, or obstacles before the rest of the body reaches them. And it makes equal sense for the nervous system's control center, the brain or ganglia, to be located near those sensory organs. The shorter the distance between sensors and decision makers, the faster the organism can react. In the time it takes for a signal to travel from the tail of an animal to its brain and back, a predator might strike, or prey might escape. By clustering sensors and nerves at the front end, bilateral animals gain a crucial advantage in speed of response. This isn't just theoretical. Studies of animal movement and behavior consistently show that bilaterally symmetrical organisms react faster and move more purposefully than their radially symmetrical or asymmetrical cousins. A bilaterally symmetrical predator, like a shark, can track down prey with precision, using its paired sensors to locate the source of vibrations or smells. A radially symmetrical jellyfish, by contrast, drifts more passively, capturing whatever happens to touch its tentacles. Even among bilaterally symmetrical animals, we can see variations in how symmetry is expressed, and these variations tell us something about lifestyle and ecology. Fish that live in open water tend to have more perfectly symmetrical bodies than fish that live near the bottom. Flatfish, like flounder, start life bilaterally symmetrical, but as they mature, one eye migrates to join the other on one side of the head, and the fish begins to live lying on its side on the seafloor. This is a rare example of fish abandoning bilateral symmetry in adulthood, an adaptation to their peculiar lifestyle. Bilateral symmetry also influences reproduction. Many bilaterally symmetrical animals have paired reproductive organs, one on each side of the body. This redundancy provides a kind of insurance. If disease or injury affects one side, the other can still function. The same principle applies to other paired organs like kidneys in vertebrates. Symmetry builds in backup systems, increasing an organism's chances of surviving long enough to reproduce. In insects, bilateral symmetry allows for incredibly complex behaviors despite relatively simple brains. An ant has a tiny nervous system compared to a mammal, but by organizing that system symmetrically, with paired sensors and paired legs, the ant can navigate complex terrain, locate food, and return to its colony. The ant's bilateral body plan makes these behaviors possible with minimal neural hardware. When we look at the fossil record, we see that bilateral symmetry appears quite suddenly in geological terms, during a period known as the Cambrian explosion, roughly 540 million years ago. Before this time, most animals were simple, often radially symmetrical or asymmetrical. Then, in a relatively short span of perhaps 10 to 20 million years, a huge diversity of bilaterally symmetrical animals appeared. These included the ancestors of almost all modern animal groups. Why did bilateral symmetry spread so rapidly? Most scientists believe it's because bilateral symmetry enabled active predation. Before the Cambrian, most animals were filter feeders or grazers, passively consuming whatever they could reach. Bilateral symmetry allowed animals to hunt, to pursue prey actively. And once some animals became active predators, there was strong selective pressure for potential prey to also become bilaterally symmetrical and mobile, to escape those predators. An evolutionary arms race began, and bilateral symmetry was the ticket to participation. This arms race drove the rapid diversification of body plans. Different groups of bilaterally symmetrical animals evolved different solutions to the challenges of predation and defense. Some became fast swimmers, others developed hard shells, still others burrowed into sediment. But nearly all of them retained bilateral symmetry as their fundamental body plan, because it provided the foundation for all these different strategies. Today, when we examine the genetics underlying bilateral symmetry, we find that many of the same genes are involved across widely different animal groups. Genes that control the development of the front and back ends, the top and bottom, and the left and right sides are remarkably similar in flies, worms, sea urchins, and mammals. This suggests that bilateral symmetry evolved once in a common ancestor of all these groups and has been maintained ever since because it works so well. The power of bilateral symmetry lies not just in what it enables, but in how it constrains and channels evolution. Once an organism has a bilaterally symmetrical body plan, certain types of adaptations become easier and others become harder. It's easier to evolve longer legs, or faster moving fins, or better eyes. It's harder to evolve additional body segments in the middle of the body, or to rearrange the positions of major organs. Bilateral symmetry acts as both an enabler and a constraint, shaping the possible paths of evolution. And yet, within those constraints, the diversity is astounding. From the sleek, streamlined dolphin to the clumsy, armored armadillo, from the delicate dragonfly to the powerful eagle, all are variations on the same basic bilateral theme. This theme has proven flexible enough to work in every environment on Earth, from the deepest oceans to the highest mountains, from frozen tundra to burning deserts. As we watch a dog running, a bird flying, or a fish swimming, we're watching bilateral symmetry in action. We're seeing the result of over 500 million years of evolution, of natural selection sculpting and refining this body plan, until it could support the incredible diversity of life we see today. And we're seeing too how form and function intertwine, how the simple principle of mirror image balance becomes the foundation for everything from speed to intelligence. Cephalization, where symmetry meets intelligence. Now let's explore one of the most profound consequences of bilateral symmetry: the evolution of heads, brains, and the concentration of intelligence at one end of the body. This process is called cephalization, from the Greek word kephale, meaning head. It's an evolutionary trend that links symmetry to consciousness itself. Imagine you're a simple worm-like creature living 500 million years ago. You have bilateral symmetry, which means you have a front and a back. As you burrow through the muddy sediment searching for food, your front end encounters obstacles, chemical signals, potential meals, and possible dangers before your back end does. Over generations, natural selection begins to favor individuals who have more sensory cells clustered at their front end. These individuals can detect food or danger a fraction of a second sooner, and that tiny advantage makes all the difference. But sensing isn't enough. You also need to process that information and respond quickly. So alongside the sensory cells, nerve cells begin to concentrate at the front end too. First, perhaps, just a simple cluster of neurons, called a ganglion. Then, as complexity increases, multiple ganglia merge and specialize, dividing labor among different types of processing. Eventually, this cluster becomes substantial enough to deserve its own name, a brain. This is cephalization. It's the gradual, evolutionary concentration of nervous and sensory tissue toward the anterior end of an organism. And it happens not once, but repeatedly across different animal lineages. Flatworms, annelids, arthropods, mollusks, and vertebrates all show cephalization, though they achieved it independently. The fact that cephalization evolved multiple times tells us it's not some quirky accident, but rather a powerfully advantageous adaptation. Let's start with one of the simplest examples. Flatworms, members of the phylum Platyhelminthes. These small creatures, often found gliding across rocks in streams or inhabiting the bodies of other animals as parasites, represent an early stage of cephalization. A flatworm has a distinct head region with simple eyes called ocelli. These eyes can't form detailed images, but they can detect light and dark, helping the worm orient itself and avoid bright, exposed areas where predators might lurk. Beneath those primitive eyes, the flatworm has a small brain, really just two ganglia fused together, connected to nerve cords that run the length of its body. This isn't much, neurologically speaking, but it's a huge leap forward from having nerves diffusely scattered throughout the body, as in animals like jellyfish. With a centralized processing center, the flatworm can integrate information from its sensors and coordinate responses more effectively. As we move to more complex animals, we see cephalization taken to greater extremes. Annelids, the segmented worms that include earthworms and leeches, have more developed brains and sensory structures. An earthworm has a cerebral ganglion at its front end that receives information from sensory cells scattered over its skin. The earthworm's brain is still quite simple compared to ours, but it's sophisticated enough to allow the worm to learn. Earthworms can be trained in laboratory experiments to navigate simple mazes, demonstrating that even modest cephalization enables basic learning and memory. Arthropods take cephalization even further. In insects, spiders, and crustaceans, the head is a distinct, heavily armored structure packed with sophisticated sense organs and a relatively large brain. Consider a honeybee. Its head houses 5 eyes, 2 large compound eyes, and 3 smaller simple eyes, plus 2 antennae covered with chemical and tactile sensors. The bee's brain, though tiny by our standards, contains about 1 million neurons, enough to support remarkably complex behaviors, including navigation, communication through dance, and social organization within the hive. The arthropod head is so important that during their evolution, multiple body segments fused together to form it. The ancestors of modern arthropods were more worm-like, with a chain of similar segments running front to back. Over time, the frontmost segments merged, their exoskeletons fusing into a single head capsule that protected the brain and concentrated the sensory organs. This fusion represents a major evolutionary investment in cephalization. Among mollusks, we see an interesting range of cephalization. Clams and oysters, which are bivalves, have very little cephalization. They're filter feeders that spend most of their lives buried in sediment or attached to rocks. They have simple sensory cells scattered around their bodies, but no real head, and certainly no brain. For their lifestyle, which involves staying in one place and straining food particles from water, heavy investment in a head and brain would be wasteful. But look at cephalopods, the group that includes octopuses, squid, and cuttlefish, and you see cephalization at its most spectacular among invertebrates. These mollusks are active predators, pursuing prey through open water. An octopus has a large, complex brain that wraps around its esophagus. It has highly developed eyes that rival vertebrate eyes in their sophistication, capable of forming detailed images and distinguishing colors. Its tentacles are covered with hundreds of chemoreceptors and touch sensors. Octopuses can solve problems, open jars, recognize individual humans, and even use tools. They can change the color and texture of their skin in milliseconds to blend into their surroundings, or communicate with other octopuses. This intelligence is made possible by their advanced cephalization. Their brain coordinates inputs from multiple sensory systems and generates complex, flexible behaviors. An octopus represents an independent evolutionary path to intelligence, one that shows how cephalization, when taken far enough, can produce sophisticated cognition, even in an animal whose last common ancestor with us lived over 500 million years ago. Then we come to vertebrates, our own group. Vertebrates include fish, amphibians, reptiles, birds, and mammals. What unites us is a backbone, yes, but also an exceptional degree of cephalization. The vertebrate head is a marvel of evolutionary engineering, housing a brain protected by a skull, eyes in precisely positioned sockets, ears with intricate internal structures, and a nose or nostrils connected to sophisticated chemical detection systems. The vertebrate brain itself is divided into regions with specialized functions. The cerebrum handles conscious thought and sensory processing. The cerebellum coordinates movement and balance. The brainstem controls automatic functions like breathing and heart rate. As vertebrates evolved and diversified, some lineages developed increasingly large and complex brains relative to their body size. In mammals, and especially in primates, brain size reached new peaks. The human brain, with its roughly 86 billion neurons, represents perhaps the most extreme example of cephalization in Earth's history. Our massive brains consume about 20% of our body's energy when we're at rest, despite accounting for only about 2% of our body weight. This is a huge metabolic cost, but it's worth it because of what our brains enable: language, abstract thought, planning for the future, complex tool use, art, science, culture. But cephalization isn't just about brain size. It's also about the integration of sensory systems with the nervous system. In vertebrates, nearly all the major sense organs are located in the head, positioned around the brain. Your eyes, ears, nose, tongue all send signals to your brain along relatively short neural pathways. This minimizes the time between sensing something and consciously perceiving it. Consider what happens when you hear a sound. Vibrations in the air cause your eardrum to vibrate. Tiny bones in your middle ear amplify these vibrations and transmit them to the cochlea in your inner ear. Hair cells in the cochlea convert the vibrations into electrical signals. These signals travel along the auditory nerve to your brain, a distance of just a few centimeters. Your brain processes the signals, and you become consciously aware of the sound. This entire process takes about 50 to 70 milliseconds, depending on the complexity of the sound. Now, imagine if your ears were located on your feet instead of your head. The neural signals would have to travel a much longer distance to reach your brain, perhaps a meter or more. At the speed neural signals travel, that extra distance would add delays of tens of milliseconds. In a dangerous situation where a fraction of a second might determine whether you escape a predator or become its meal, those extra milliseconds could be fatal. Cephalization, by keeping sensors close to the brain, minimizes these delays. The evolution of cephalization in vertebrates also involved changes to the skeleton. The skull evolved to protect the brain, a unique structure made of multiple bones that fuse together as an individual develops. The vertebrate skull is flexible at birth, allowing the head to pass through the birth canal in mammals, but becomes rigid in adulthood to shield the brain from injury. This protective housing allows for the development of larger, more complex brains without increasing vulnerability. In many vertebrates, cephalization has been linked to the evolution of predation. Active predators generally have more cephalized bodies than passive filter feeders or grazers. A shark has acute senses and a brain capable of tracking prey across large distances. A hagfish, which mostly scavenges dead animals, has much less cephalization. The correlation isn't perfect, but the pattern is clear. Hunting requires intelligence, and intelligence requires cephalization. Interestingly, some animal groups have lost cephalization after evolving it. Echinoderms, which include sea stars and sea urchins, evolved from bilaterally symmetrical, cephalized ancestors. But, as adults, they display radial symmetry and have no distinct head. Their nervous system is distributed in a ring around their body, with nerve cords running into each arm. This represents a secondary loss of cephalization, likely related to their adoption of a slower, more sedentary lifestyle. Bivalve mollusks, like clams, show a similar pattern. Their ancestors were likely cephalized, as seen in the close relatives like snails. But as bivalves evolved to filter feed in sediment, they lost their heads and their brains, retaining only scattered ganglia. This shows that cephalization, while advantageous in many contexts, is not universally beneficial. For organisms that don't move much and don't need to process complex sensory information quickly, the metabolic cost of maintaining a brain may outweigh the benefits. For those of us with advanced cephalization, though, the benefits are undeniable. Your ability to read these words, to understand them, to form mental images of the concepts they describe, all of this depends on your cephalized nervous system. Your brain receives input from your eyes, compares it to stored memories, generates meaning, and produces conscious experience. This extraordinary capability is the endpoint of hundreds of millions of years of evolution, starting with simple clusters of neurons in the front end of worm-like ancestors. Cephalization reminds us that intelligence isn't separate from the physical structure of bodies. Our minds emerge from the arrangement and activity of neurons in our brains, and our brains exist in their current form because of cephalization. The concentration of nervous tissue at one end of a bilaterally symmetrical body was a necessary step on the path from simple organisms to complex, conscious beings. As we go about our days, using our senses to navigate the world, our brains to solve problems, our heads to hold conversations. We're experiencing the legacy of cephalization. We're living proof of how an evolutionary trend that began with primitive worms has culminated in creatures capable of understanding their own existence, of wondering about their place in the universe, and of studying the very processes that brought them into being. Radial symmetry, the circle of life. Now, let's turn our attention to a different kind of pattern, radial symmetry. If bilateral symmetry is the architecture of movement and intelligence, radial symmetry is the geometry of patience and reception. It's the design that says, "The world comes to me from all directions, and I am equally prepared." Radial symmetry means that an organism's body parts are arranged around a central axis, like spokes radiating from the hub of a wheel. You can slice through a radially symmetrical creature at any angle through its central point, and you'll get roughly matching halves. These organisms have a top and bottom, an oral surface where the mouth is located, and an aboral surface opposite it, but they have no distinct left, right, front, or back. The most familiar examples of radial symmetry come from the ocean. Jellyfish, with their bell-shaped bodies and tentacles trailing below, display radial symmetry. Sea anemones, those flower-like creatures that attach to rocks in tidal zones, are radially symmetrical. So are corals, the tiny colonial animals that build vast reef structures. All of these belong to a phylum called Cnidaria, and all share the basic body plan of radial symmetry. When you watch a jellyfish pulsing through the water, you're watching radial symmetry in motion. The jellyfish's bell contracts evenly around its entire circumference, pushing water downward and propelling the animal upward. Its tentacles hang down in a roughly circular arrangement, ready to capture any small animal that brushes against them from any direction. The jellyfish doesn't hunt actively, pursuing specific prey. Instead, it drifts with currents, and its radial symmetry ensures that it can respond to prey or danger coming from anywhere. This is the key advantage of radial symmetry. It suits organisms that are either sessile, meaning they stay in one place, or that float passively with water currents. A sea anemone attached to a rock doesn't know which direction food will come from. Plankton might drift past from any angle. Radial symmetry, with its ring of tentacles surrounding the mouth, provides equal coverage in all directions. The anemone doesn't need to turn or orient itself. It's always ready, always facing everywhere at once. Even the cnidarians that do move tend to move slowly and without much directionality. A jellyfish mostly drifts, using its pulsing movement primarily to maintain vertical position in the water column rather than to travel horizontally. Some jellyfish can swim toward light or away from obstacles, but these movements are sluggish compared to the darting, purposeful swimming of bilaterally symmetrical fish. The internal anatomy of radially symmetrical animals reflects their symmetry. Inside a jellyfish's bell, you'll find a gastrovascular cavity, a simple stomach that also serves as a circulatory system. Radial canals extend from this central cavity out toward the edge of the bell, distributing nutrients in a pattern that mirrors the animal's external symmetry. There's no complex system of organs because there's no need for one. The jellyfish's lifestyle doesn't require the kind of specialization you see in bilaterally symmetrical animals. Radial symmetry also tends to correlate with simpler nervous systems. Cnidarians don't have brains or concentrated nerve ganglia. Instead, they have a diffuse nerve net, a web of neurons spread throughout their tissues. When a jellyfish's tentacle touches something, signals spread through the nerve net, causing nearby muscle cells to contract. There's no central processing, no decision-making hub. The response is local and reflexive. This might seem primitive, and in some ways it is. But for the jellyfish's lifestyle, it works perfectly. A nerve net can respond quickly to stimuli from any direction, without the delays that would come from routing signals through a central brain. When you're a passive drifter, reacting instantly to whatever touches you is more important than having complex cognition. Let's explore another group of radially symmetrical animals, ctenophores, also known as comb jellies. These beautiful creatures superficially resemble jellyfish, but they're actually quite different and belong to their own phylum. Ctenophores are named for the rows of cilia, tiny hair-like structures that run along their bodies. These cilia beat in coordinated waves, refracting light into shimmering rainbows as the comb jelly swims. Most ctenophores display biradial symmetry, a variation of radial symmetry where the body can be divided into matching halves along only two planes, rather than infinitely many. Despite this slight modification, ctenophores still fit the radial lifestyle. They're mostly planktonic, drifting with currents, capturing prey with sticky tentacles or specialized colloblasts. The existence of biradial symmetry in ctenophores might represent an intermediate evolutionary stage. Some scientists hypothesize that bilateral symmetry evolved from radial symmetry through a biradial intermediate stage. According to this theory, radially symmetrical ancestors first developed biradial symmetry with two special planes of symmetry, and then eventually transitioned to full bilateral symmetry with only one plane of symmetry. This progression would make sense if we think of evolution as a series of small steps rather than giant leaps. Radial symmetry isn't limited to animals. In the plant kingdom, many flowers display radial symmetry, though botanists use the term actinomorphic instead. An actinomorphic flower has petals, sepals, and other parts arranged in a circular pattern around its center. You can rotate the flower and it looks the same at multiple angles. Lilies, buttercups, and roses are all actinomorphic. Why would a flower evolve radial symmetry? For similar reasons as an animal might. A radially symmetrical flower can be approached by pollinators from any angle. A bee doesn't need to land on a specific side or orient itself in a particular way. It can land anywhere on the flower and still access the nectar and pollen. This generalist approach works well for flowers that are pollinated by many different types of insects. In the fossil record, we see evidence that radial symmetry is ancient. Some of the earliest multicellular animals, dating back over 600 million years to the Ediacaran period, appear to have been radially symmetrical. These strange, flat creatures left impressions in seafloor sediments, and many of those impressions show circular, radiating patterns. We don't know exactly what these organisms were or how they relate to modern animal groups, but their radial symmetry suggests a lifestyle similar to modern cnidarians, sessile or slowly drifting, interacting with the environment equally from all sides. The transition from radial to bilateral symmetry represents one of the major events in animal evolution. Exactly how and why this transition occurred remains debated. One hypothesis suggests that an ancestral radially symmetrical organism adopted a crawling lifestyle, moving along the seafloor. Once it started moving consistently in one direction, natural selection began to favor individuals with slight asymmetries that made directional movement more efficient. The end that led the way became differentiated from the trailing end, and bilateral symmetry was born. Another hypothesis suggests that bilateral symmetry evolved in response to burrowing. Many early bilaterally symmetrical animals were worm-like creatures that tunneled through sediment. Burrowing imposes strong directional constraints. You dig forward through the substrate. A radially symmetrical body plan wouldn't work well for burrowing. A bilaterally symmetrical, elongated body would be much more efficient. Once bilateral symmetry proved advantageous for burrowing, it could then be co-opted for other forms of locomotion. Regardless of how the transition happened, the result was dramatic. The Cambrian explosion, that burst of diversification roughly 540 million years ago, saw bilateral animals rise to dominance. Radially symmetrical animals didn't disappear. Cnidarians and ctenophores persist in huge numbers in modern oceans, but they were joined and eventually outnumbered by their bilaterally symmetrical cousins. Today, when we encounter radially symmetrical animals, we're often drawn to their aesthetic appeal. There's something hypnotic about the circular perfection of a sea anemone's tentacles, or the pulsing translucence of a jellyfish. Artists and designers have long been inspired by radial symmetry, incorporating it into mandalas, rose windows in cathedrals, and decorative patterns in textiles and architecture. This appeal might stem from the fact that radial symmetry is relatively rare in our everyday experience. Most of the animals we interact with regularly, pets, livestock, wildlife, are bilaterally symmetrical like us. Encountering radial symmetry feels novel, exotic, a reminder that life has explored many different solutions to the challenge of existence. Radial symmetry also embodies a certain kind of serenity. A jellyfish doesn't struggle or strain. It doesn't chase or flee. It simply exists, floating, pulsing, responding to whatever the ocean brings. In a world where so much of biology is about competition, about the relentless drive to survive and reproduce, radially symmetrical animals seem almost meditative in their simplicity. Of course, this is romanticizing. A jellyfish's life is no less shaped by natural selection than a shark's. It must capture enough prey to sustain itself. It must avoid predators and parasites. It must find mates and reproduce. But the way it accomplishes these tasks is fundamentally different from bilaterally symmetrical animals, and that difference is encoded in its body symmetry. As we drift through these thoughts like a jellyfish through water, we're reminded that there is no single right way to build a body. Bilateral symmetry works brilliantly for active, mobile, intelligent animals. Radial symmetry works equally well for sessile or planktonic organisms that interact with a world without clear directionality. Evolution has produced both designs because both fit particular ecological niches. Together, they demonstrate nature's versatility, the many successful solutions that have emerged from the simple laws of variation and selection. The starfish paradox, a symmetry story. Among the wonders of the ocean, few creatures capture our imagination, quite like sea stars, commonly though incorrectly called starfish. With their 5 arms radiating from a central disk, they seem to be textbook examples of radial symmetry. But the sea star holds a secret, a hidden story written in its development that challenges our neat categories and reveals the deep evolutionary history connecting different body plans. Sea stars belong to a phylum called Echinodermata, which also includes sea urchins, sand dollars, sea cucumbers, and sea lilies. Echinoderm means spiny skin, referring to the bumpy, sometimes spiny texture of these animals' surfaces. Adults of all these groups display a special form of radial symmetry called pentaradial symmetry, meaning fivefold symmetry. Count the arms of a sea star, 5. Count the sections of a sand dollar's test, its hard shell, 5. Even sea cucumbers, which look more worm-like, have 5 rows of tube feet running along their bodies. This fivefold symmetry is distinctive and consistent across the entire phylum. It's so characteristic that if you find a fossil with pentaradial symmetry, you can be confident it's an echinoderm, or an ancestor of echinoderms. The pattern has persisted for hundreds of millions of years, suggesting it offers significant advantages for these animals' lifestyles. But here's where things get interesting. If you watch an echinoderm develop from a fertilized egg, you see something unexpected. The larva that hatches is not radially symmetrical at all. It's bilaterally symmetrical. Echinoderm larvae are tiny, transparent creatures that swim through the plankton using bands of cilia. They have a distinct front and back, left and right. They're organized along the same body plan as fish larvae, or tadpoles, or human embryos. This is remarkable. It means that at some point in echinoderm evolution, a bilaterally symmetrical ancestor gave rise to radially symmetrical descendants. Echinoderms didn't start with radial symmetry. They evolved it secondarily, switching from one body plan to another. The bilaterally symmetrical larvae are ghosts of that ancestral form, developmental echoes of an earlier time. The transformation from bilateral larva to radial adult happens during metamorphosis. As the larva develops, parts of its left side begin to grow and expand, while the right side is gradually reabsorbed. The left side essentially swallows the right side, then reorganizes itself into a pentaradial pattern. It's one of the most dramatic metamorphoses in the animal kingdom, a complete restructuring of body symmetry. Why would evolution do this? Why take a perfectly good bilateral body plan and transform it into something radial? The answer lies in lifestyle. The ancestors of modern echinoderms were likely active, mobile, bilaterally symmetrical creatures. But at some point, perhaps 600 million years ago, some of these creatures adopted a more sedentary existence. Maybe they began attaching themselves to rocks or the seafloor, or moved very slowly while feeding on detritus or small organisms. For a slow-moving or sessile organism, radial symmetry offers advantages. You don't need to turn your body to face a particular direction. You can feed or sense danger from any angle equally well. The energetic cost of maintaining directional movement systems, streamlined body shapes, concentrated sensory organs at one end becomes unnecessary. Radial symmetry is more metabolically efficient for this lifestyle. But evolution can't just throw away the accumulated adaptations of millions of years. The developmental programs that build bilateral larvae are deeply embedded in echinoderm DNA, controlled by ancient genes that also function in other bilaterally symmetrical animals. So echinoderms retain bilateral symmetry during early development, then transition to radial symmetry as they mature and adopt their adult lifestyle. Interestingly, research suggests that adult echinoderms may retain more bilateral organization than is obvious from their external appearance. Studies of sea star behavior have found that they tend to have a preferred arm, one they lead with when crawling. If you number a sea star's arms 1 through 5, arm 5 is often the one that points forward during movement. The arms on either side of arm 5, arms 4 and 1, tend to move in coordination, while arms 2 and 3 trail behind. This creates a rough bilateral pattern of movement despite the animal's pentaradial anatomy. Genetic studies support this finding. Sea stars possess many of the same genes that control bilateral symmetry in other animals, including genes from the Hox family, which specify the front-to-back axis in bilaterally symmetrical creatures. These genes are still active in sea stars, and they're expressed in patterns that suggest an underlying bilateral organization, with arm 5 representing the anterior or front end, and the space between arms 2 and 3 representing the posterior or back end. This hidden bilaterality has important implications. It suggests that the transition from bilateral to radial symmetry in echinoderms wasn't complete. Instead, radial symmetry is superimposed on an underlying bilateral scaffold. The adult sea star has 5 arms arranged symmetrically, but genetically and behaviorally, it still knows which direction is forward. From an evolutionary perspective, this makes sense. Evolution is a tinkerer, not an engineer starting from scratch. It works with existing structures and programs, modifying them in incremental steps. Completely erasing all traces of bilateral symmetry would require extensive genetic changes. It's easier and less disruptive to simply add a new layer, radial symmetry in adulthood, while retaining the bilateral foundation. The echinoderm story reminds us that animal symmetry isn't always what it appears to be. What looks like straightforward radial symmetry in an adult sea star is actually a complex overlay on a bilaterally symmetrical genetic and developmental program. This layering of body plans, one on top of another, is a hallmark of evolutionary change. Evolution rarely makes clean breaks with the past. It preserves what works and modifies it to fit new circumstances. Other echinoderms show variations on this theme. Sea cucumbers, for example, have re-evolved a kind of bilateral symmetry in their adult forms. Unlike sea stars, which crawl with their oral surface facing downward, sea cucumbers lie on their sides and crawl along their length. This horizontal orientation reintroduces bilateral organization. They have a distinct top and bottom, a front and back. Yet, they still retain pentaradial symmetry in the arrangement of their tube feet and other structures. They're bilateral and pentaradial at the same time. Sea urchins take the pentaradial pattern to an extinct extreme, with their spherical bodies covered in spines. But even in sea urchins, developmental biologists have found evidence of underlying bilateral organization. The pattern of gene expression during sea urchin development shows bilateral features, and some species of sea urchin larvae are more clearly bilateral than others. The echinoderm case study illustrates a broader principle. Body symmetry is developmentally flexible. It's not rigidly determined from the very start of an organism's life. Instead, it can change during development, and over evolutionary time, lineages can switch between different symmetry types. This flexibility has allowed animals to adapt to wildly different ecological niches, while still maintaining some continuity with their evolutionary past. For us, watching a sea star slowly crawl across a tidal pool, there's something profound in knowing that this creature embodies a transformation, a shift from one way of organizing a body to another. Its 5 arms speak of a lifestyle adapted to slow movement, to interacting with the world from all sides, to a benthic existence on the seafloor. But its larval past and its hidden bilateral tendencies speak of an earlier time when its ancestors swam actively through ancient seas, bilaterally symmetrical and cephalized. The starfish paradox teaches us to look deeper than surface appearances. It shows us that evolution is a historical process, carrying forward the marks of past forms even as it creates new ones. And it reminds us that the categories we create, bilateral, radial, pentaradial, are useful for understanding nature, but nature itself doesn't always fit neatly into our boxes. Instead, it blends and layers and recombines patterns in endlessly creative ways. Flower symmetry, the art of attraction. While we've been focusing on animals, let's not forget that symmetry shapes the plant kingdom too, especially in that most celebrated of plant structures, the flower. Flowers exist to reproduce, and their shapes, colors, and symmetries are all evolutionary solutions to a single challenge: how to get pollen from one plant to another of the same species. And in this arena, symmetry plays a starring role. Most flowers display what botanists call actinomorphic symmetry, also known as radial symmetry. Just like a sea anemone, an actinomorphic flower has parts arranged in a circle around its center. You can rotate the flower, and it looks essentially the same at multiple angles. Roses, lilies, buttercups, and sunflowers are all actinomorphic. If you were to draw lines through the center of these flowers, you'd find that many such lines could divide the flower into matching halves. Actinomorphic flowers are the ancestral condition for flowering plants. The earliest flowers, appearing in the fossil record around 140 million years ago, were likely radially symmetrical. This makes sense from a pollination perspective. Early flowers were probably pollinated by generalist insects, beetles, and flies that weren't particularly specialized in their flower-visiting behavior. For these pollinators, a radially symmetrical flower is ideal. The insect can land anywhere on the flower and still access the nectar and pollen. The flower doesn't need to guide the insect to a specific position. But later in flowering plant evolution, something remarkable happened. Some flowers evolved a different kind of symmetry, zygomorphic symmetry, also known as bilateral symmetry. A zygomorphic flower can be divided into matching halves along only one plane, usually running from top to bottom through the center. One side mirrors the other, but if you rotate the flower, the pattern changes. Orchids, snapdragons, peas, and many members of the mint family have zygomorphic flowers. The evolution of zygomorphic flowers coincides with the diversification of specialized pollinators, particularly bees. Unlike the generalist insects that pollinated early flowers, bees are highly adapted for flower visiting. They can learn to associate specific flower shapes, colors, and scents with nectar rewards, and zygomorphic flowers take advantage of this learning ability. A zygomorphic flower often has a landing platform, usually formed by modified petals, where a bee can comfortably land. The flower's shape then guides the bee to a specific position where it will brush against the stamens, picking up pollen, or against the stigma, depositing pollen. This precision greatly increases the efficiency of pollination. Instead of pollen being scattered randomly on the bee's body, it's placed deliberately on specific parts of the bee, usually the head or back, where it won't accidentally rub off until the bee visits another flower of the same species. Think of an orchid. Many orchids have elaborate zygomorphic flowers with a specialized lower petal called a lip or labellum. that serves as a landing pad. The rest of the flower is structured like a tunnel or chamber. When a bee lands on the lip and crawls inside seeking nectar, the flower's shape forces the bee into a specific orientation. The pollen masses, called pollinia, are positioned so they stick to the bee's head or back. When the bee visits another orchid, these pollinia are precisely positioned to contact the stigma. This specificity benefits both plant and pollinator. The plant ensures that its pollen goes to others of its species, rather than being wasted on the wrong species. The bee, meanwhile, becomes more efficient at harvesting nectar from this type of flower. Over time, this can lead to coevolution, where the flower shape and the bee's behavior and body shape evolve together, each influencing the other. But zygomorphy comes with trade-offs. Studies have shown that flowers with bilateral symmetry tend to attract fewer different species of pollinators than radially symmetrical flowers. A zygomorphic flower is specialized, tailored to a particular type of pollinator, usually bees. This specialization means fewer kinds of insects visit the flower, because many potential pollinators are either physically unable to access the flower's rewards or haven't learned how to manipulate the flower properly. This narrow pollinator base can be risky. If the preferred pollinator declines in numbers due to disease, habitat loss, or climate change, the zygomorphic flower has few backup options. Actinomorphic flowers, visited by many different types of insects, are more resilient to pollinator loss. This is one reason why actinomorphic flowers remain common despite the advantages of zygomorphy. Research using plant-pollinator interaction networks has confirmed these patterns. In one large study examining thousands of plant species and their pollinators, scientists found that zygomorphic flowers had about 4 visitor species on average, while actinomorphic flowers had 5. That might not sound like a huge difference, but in ecological terms, it can be significant. Fewer pollinator species mean less redundancy, higher vulnerability to disruption. At the same time, that study found that zygomorphic flowers shared more of their pollinators with each other than actinomorphic flowers did. In other words, the bees that visit one zygomorphic flower are likely to visit other zygomorphic flowers in the area. This makes sense. A bee that has learned to manipulate complex flower shapes has a skill that transfers to other similarly shaped flowers. So while zygomorphic flowers may have fewer total pollinators, those pollinators are often specialists that visit many zygomorphic species. Interestingly, recent research suggests that zygomorphic flowers actually remain open and functional for longer periods than actinomorphic flowers. This longer flowering time might compensate for their fewer pollinators. If you have fewer visitors, you need to give them more time to find you. By staying open longer, zygomorphic flowers increase their chances of being pollinated despite their narrower pollinator base. The symmetry of a flower can even affect how much variation there is in flower size within a population. Theory predicts that zygomorphic flowers should show less size variation than actinomorphic flowers because specialized pollinators exert stabilizing selection. If a flower is too large or too small, it might not fit the pollinator properly, leading to inefficient pollen transfer. Actinomorphic flowers, visited by many different sized pollinators, face no such constraint. Some studies have found support for this hypothesis, with zygomorphic flowers showing less size variation. But other studies have found the opposite, or no difference, suggesting that the relationship between symmetry and size variation is more complex than simple theory predicts. Factors like whether the plant can self-pollinate and how ecologically specialized it is also play roles. The genetic basis of flower symmetry has been extensively studied, particularly in snapdragons, a classic zygomorphic flower. Scientists have identified a gene called cycloidea that plays a key role in establishing bilateral symmetry in snapdragon flowers. When cycloidea is active in the developing flower, it causes the upper petals to develop differently from the lower petals, creating the characteristic two-sided symmetry. If you disable cycloidea through mutation, the flower develops with radial symmetry instead. All the petals become similar, arranged in a ring. This transformation is called peloria, and it occasionally occurs spontaneously in wild snapdragon populations. Charles Darwin was fascinated by peloric snapdragons and described them in his work on variation in domesticated plants. The discovery that a single gene can switch a flower between radial and bilateral symmetry reveals how evolution can make dramatic changes through relatively simple genetic modifications. It also suggests that the evolution of zygomorphy from actinomorphy might not require extensive genetic rewiring. Changing the expression of a few key genes might be sufficient. This has happened repeatedly in flowering plant evolution. Zygomorphic flowers have evolved independently in many different plant families, suggesting that the transition from radial to bilateral symmetry is relatively easy, evolutionarily speaking. Once the genetic machinery for making petals exists, repurposing that machinery to make different types of petals in different positions isn't a huge leap. In gardens and wildflower meadows, we can see the consequences of these evolutionary processes playing out. Radially symmetrical flowers like daisies are visited by a wide variety of insects: bees, flies, butterflies, beetles. Bilaterally symmetrical flowers like lavender or sage are visited primarily by bees, which have learned to land on the lower lip and push their way into the flower tube to reach the nectar inside. For a flower, symmetry isn't just an aesthetic choice. It's a functional strategy with deep implications for reproduction. The symmetry of a flower determines which animals will pollinate it, how efficiently pollen transfer occurs, how vulnerable the plant is to pollinator loss, and ultimately, how successful the plant will be at producing seeds and spreading its genes. And for us, observers of this intricate dance between plants and pollinators, flower symmetry offers a window into evolution's creativity. It shows us how a single trait, the symmetry of a flower, can shape ecological relationships, influence which species thrive and which decline, and even drive the diversification of entire groups of organisms. In every garden, in every wildflower field, symmetry is silently shaping the relationships between plants and the insects that visit them, a testament to the power of small structural changes to have profound effects. The mathematics of beauty, Fibonacci and fractals. So far, we've explored symmetry as a concept of balance and repetition in space. But there's another layer to nature's patterns, one that involves numbers, ratios, and infinite recursion. This is the realm of the Fibonacci sequence and fractal geometry, where mathematical principles generate shapes of stunning complexity and beauty. The Fibonacci sequence is deceptively simple. It begins with 0 and 1, and each subsequent number is the sum of the two preceding numbers. So the sequence goes 0, 1, 1, 2, 3, 5, 8, 13, 21, 34, 55, and so on, extending infinitely. This sequence was first described by Leonardo Fibonacci, an Italian mathematician, in the 13th century, though earlier mathematicians in India had discovered it even before him. What makes the Fibonacci sequence remarkable isn't just its elegant simplicity. It's that this sequence appears over and over again in nature. The number of petals on many flowers follows Fibonacci numbers. Lilies have 3 petals, buttercups have 5, marigolds have 13, asters have 21, and daisies can have 34, 55, or even 89 petals. Not every flower follows this pattern, but enough do that it's clearly not coincidence. The spirals in a sunflower seed head follow Fibonacci numbers too. If you look closely at the center of a sunflower, you'll see 2 sets of spirals, one curving clockwise, the other counterclockwise. Count the number of spirals in each direction, and you'll typically find consecutive Fibonacci numbers. A common sunflower might have 34 spirals in one direction and 55 in the other, or 55 and 89. Why does this happen? The answer lies in a number closely related to the Fibonacci sequence called the golden ratio, represented by the Greek letter phi, approximately equal to 1.618. If you divide any Fibonacci number by the one before it, you get a ratio that gets closer and closer to phi as you move further along the sequence. 21 divided by 13 gives about 1.615. 34 divided by 21 gives about 1.619. The larger the numbers, the closer the ratio gets to the golden ratio. The golden ratio appears when you try to pack seeds or leaves or flower petals as efficiently as possible. Imagine you're a plant and you're producing seeds in the center of your flower head. Each seed forms at a certain angle from the previous seed. If that angle is based on the golden ratio, specifically 360 degrees divided by phi squared, the seeds will pack together in a spiral pattern that minimizes gaps and maximizes space efficiency. This is exactly what we see in sunflowers and pine cones. The same principle applies to the arrangement of leaves on a stem, a phenomenon called phyllotaxis. Many plants arrange their leaves in spiral patterns around the stem, with each leaf positioned at an angle from the previous one. When this angle is based on the golden ratio, the leaves end up distributed evenly around the stem, ensuring that upper leaves don't block sunlight from reaching lower leaves. This maximizes each leaf's access to sunlight for photosynthesis. The Fibonacci sequence also shows up in the branching patterns of trees. A tree trunk might split into 2 main branches. Each of those branches might split into 2 more, but not usually at the same time. So you get a branching pattern that follows a Fibonacci-like progression. 1 trunk, 2 main branches, 3 secondary branches, 5 tertiary branches, and so on. This branching strategy balances structural stability with efficient distribution of nutrients and water. Now, let's move from Fibonacci numbers to fractals. A fractal is a geometric shape that displays self-similarity at different scales. Zoom in on part of a fractal, and you see the same basic pattern repeated. Zoom in further, and you see it again. This recursive repetition can continue infinitely. Nature is full of fractals. The branching of trees is fractal. The pattern of large branches splitting into smaller branches, which split into even smaller branches, repeating down to the finest twigs. River networks are fractal. Main rivers fed by tributaries, which are fed by smaller streams, which are fed by tiny rills, all following similar branching patterns. Coastlines are fractal. The jagged outline of a continent contains bays and peninsulas, which contain smaller inlets and promontories, which contain still smaller irregularities, right down to the individual pebbles on a beach. Your own lungs are fractal. The trachea branches into two main bronchi, which branch into smaller bronchi, which branch into bronchioles, which branch into even smaller airways, finally ending in millions of tiny alveoli where gas exchange occurs. This fractal branching packs an enormous surface area into a relatively small volume, allowing efficient oxygen absorption. Blood vessels form fractal networks too. Your aorta branches into major arteries, which branch into smaller arteries, which branch into arterioles, which branch into capillaries. The same pattern in reverse carries blood back to your heart through venules and veins. This fractal architecture ensures that blood reaches every cell in your body, despite the limited space available. Fractals emerge from simple iterative processes. You start with a simple shape, apply a rule to it, then apply the same rule to the result, and keep repeating. In biological systems, these rules are encoded in genes and developmental processes. A tree's branching pattern results from cells at branch tips responding to hormones and growth factors. When the concentration of these signals reaches a certain threshold, the growing tip splits into two new tips, each of which can potentially split again later. The rule is simple, but repeated thousands of times. It creates the complex fractal structure of a mature tree. The connection between fractals and the Fibonacci sequence becomes clear when you look at spirals. The logarithmic spiral, where the distance from the center increases exponentially as you move around the spiral, often incorporates the golden ratio. Each quarter turn of the spiral expands the radius by a factor related to phi. This spiral appears in nautilus shells, in the curve of hurricane clouds, in the arms of spiral galaxies. In a nautilus shell, as the animal grows, it adds new chambers to its shell. Each chamber is larger than the previous one by a constant proportion, approximately the golden ratio. The result is a logarithmic spiral, a shape that allows the nautilus to grow continuously while maintaining the same overall shell shape. This is efficient. The nautilus doesn't need to remodel old parts of its shell. It simply adds new chambers in the same proportional pattern. Hurricanes form logarithmic spirals too, though for different reasons. Here, the spiral emerges from the interaction between Coriolis forces, from Earth's rotation and the inward flow of air toward a low-pressure center. As air spirals inward, conservation of angular momentum causes it to speed up, just like a figure skater spinning faster when pulling their arms in. The result is a spiral pattern that, when photographed from space, often approximates a logarithmic spiral based on the golden ratio. Even galaxies show spiral patterns related to Fibonacci and phi. Spiral galaxies, like our Milky Way, have arms that curve outward from a central bulge. These arms aren't solid structures. They're density waves, regions where stars and gas are temporarily more concentrated. The spiral pattern persists even as individual stars orbit through it. And the shape of these spirals often follows logarithmic curves related to the golden ratio. Why does the golden ratio appear so often in nature? Part of the answer is that it represents an optimization. The golden ratio is the most irrational number in a mathematical sense. It's the number that's hardest to express as a fraction. This property makes it useful for avoiding resonances and overlaps. when plants arrange their leaves or seeds according to the golden ratio, they avoid situations where multiple structures line up, which could create gaps or inefficiencies. But there's also an element of inevitability. Given the constraints of growth, the physical laws governing how structures branch or spiral, and the mathematics of packing and tiling, certain patterns emerge repeatedly. The Fibonacci sequence and the golden ratio aren't magic. They're consequences of optimization under constraint. The universe exploring the most efficient ways to organize matter and energy. Humans have been fascinated by these mathematical patterns for thousands of years. The golden ratio appears in ancient Greek architecture in the proportions of the Parthenon. Renaissance artists like Leonardo da Vinci studied the golden ratio and incorporated it into their compositions. Modern architects and designers still use these proportions, believing they create more aesthetically pleasing forms. Is there a biological basis for this aesthetic preference? Some researchers think so. Studies have shown that humans tend to prefer visual patterns with fractal dimensions around 1.3 to 1.5. Interestingly, this is the fractal dimension of many natural scenes: trees, clouds, coastlines. We evolved in environments filled with these fractal patterns, so it makes sense that our visual systems would be tuned to find them appealing. This suggests a deep connection between mathematics, nature, and human perception. The same patterns that make a tree efficient at gathering sunlight are also patterns that humans find beautiful. The same spirals that allow seeds to pack efficiently in a flower head are also spirals that catch our eye and hold our attention. Mathematics isn't separate from nature. It's the language nature speaks, the grammar underlying all the diverse structures and forms we see around us. As we contemplate a pine cone, counting its spirals and finding Fibonacci numbers, or as we watch a fern unfold, each tiny leaflet a miniature version of the whole frond, we're witnessing mathematics in action. We're seeing how simple rules, repeated and elaborated, can generate infinite complexity and beauty. And we're reminded that pattern and order aren't imposed on nature from outside. They emerge from within, from the fundamental properties of matter and energy, from the constraints of physics and chemistry, from the relentless logic of evolution seeking efficiency and adaptation. Crystalline symmetry, the geometry of ice. Let's drift now from the warm world of plants and animals to something colder, more precise, more geometrically perfect: the snowflake. If there's an icon of symmetry in nature, it's the six-sided snowflake, that tiny ice crystal that has captivated humans for centuries, with its intricate beauty and its promise that no two are exactly alike. A snowflake begins its life high in the atmosphere when water vapor, the gaseous form of water, encounters extremely cold temperatures. At temperatures below freezing, water molecules can transition directly from vapor to solid ice, skipping the liquid phase entirely. This process is called deposition, but deposition doesn't happen spontaneously. The water molecules need a nucleation site, a tiny particle around which they can begin to organize. This particle might be a speck of dust, a grain of pollen, even a bacterium. Once a few water molecules have attached to the nucleation particle, they begin to form a crystal structure. And here's where symmetry enters the picture. Water molecules, each made of one oxygen atom bonded to two hydrogen atoms, have a specific geometry. The hydrogen atoms are arranged at an angle of about 104.5 degrees from each other. When water molecules freeze into ice, this geometry dictates how the molecules stack together. In ice, water molecules arrange themselves into a hexagonal lattice. Each molecule is surrounded by 4 others in a tetrahedral configuration. But when these tetrahedra link together in a pattern, they form layers of hexagonal rings. These hexagonal layers stack on top of each other, creating the 3-dimensional structure of ice. And this hexagonal structure at the molecular level gives rise to the 6-sided symmetry we see in macroscopic snowflakes. Think of it this way: the snowflake's shape is determined by the shape of the water molecule and the angles at which water molecules prefer to bond to each other. Its molecular symmetry expressing itself on a scale visible to the human eye. This is one of nature's most direct demonstrations of how microscopic structure determines macroscopic form. As the initial ice crystal grows, water vapor molecules from the surrounding air continue to attach to it. The crystal grows faster in some directions than others, depending on temperature and humidity. At certain temperatures, the crystal grows primarily along its edges, creating flat, plate-like snowflakes. At other temperatures, it grows more along its faces, creating columnar snowflakes. And at still other temperatures, it grows with elaborate branching structures, the classic dendritic snowflakes that people picture when they think of snowflakes. The most photogenic snowflakes, those intricate 6-armed stars, form at temperatures around -15 degrees Celsius. At this temperature, the growth rate is just right to produce branches with side branches, creating fractal-like patterns that can be astonishingly complex. Each of the 6 arms grows independently, but because all 6 arms are experiencing essentially the same atmospheric conditions, they all grow in nearly the same way, producing the snowflake's characteristic symmetry. This is the key to understanding why snowflakes are symmetrical. The snowflake is so small, typically only a few millimeters across, that the environment around it is virtually uniform. As the snowflake tumbles through the air, all 6 arms experience the same temperature, the same humidity, the same air pressure at the same time. When the temperature drops slightly, all 6 arms respond by growing a little faster. When humidity increases, all 6 arms sprout new side branches. The 6-fold symmetry of the underlying crystal lattice, combined with the uniform environment, produces a snowflake where the 6 arms mirror each other. But why isn't every snowflake identical? Because even though the environment is uniform around a single snowflake, it varies from one location to another and from one moment to the next. As a snowflake falls through the atmosphere, it passes through regions with different temperatures and humidity levels. It might form in one layer of the cloud, fall into a warmer layer where growth slows, then fall into a colder layer where growth speeds up again. Each of these transitions leaves a mark on the snowflake's structure, a change in growth pattern that's recorded in the ice. Since no 2 snowflakes follow exactly the same path through the atmosphere, and since atmospheric conditions are constantly changing in complex, chaotic ways, no 2 snowflakes end up with exactly the same history. And since their history determines their shape, no 2 snowflakes are exactly alike. Each snowflake is unique, but each is also symmetrical because the 6 arms always experience the same conditions at the same time. This combination of symmetry and uniqueness has fascinated scientists and artists alike. In the late 19th century, a farmer in Vermont named Wilson Bentley became obsessed with photographing snowflakes. He developed a technique for capturing them on glass slides and photographing them through a microscope before they melted. Over his lifetime, Bentley photographed thousands of snowflakes, documenting their incredible diversity while also noting their consistent six-fold symmetry. His work helped popularize the idea that every snowflake is unique and revealed to the world just how beautiful these tiny ice crystals can be. Modern scientists have gone further, using high-speed photography and microscopy to study snowflake formation in detail. They've discovered that there are actually many different types of snowflakes, not just the classic dendritic stars. There are simple hexagonal plates, columns, needles, and even irregular forms that don't show clear symmetry at all. The conditions of temperature and humidity determine which type forms, and scientists have created detailed diagrams mapping out which conditions produce which shapes. But the classic six-armed snowflake remains the most iconic, and for good reason. These snowflakes display both symmetry and complexity, two qualities that humans find deeply appealing. The symmetry speaks to order, to underlying structure, to the fact that the universe follows rules. The complexity speaks to diversity, to the inexhaustible creativity of natural processes, to the fact that even within rules, there is room for infinite variation. There's also something humbling about snowflakes. These structures of such perfection and intricacy form spontaneously without any conscious design, through nothing more than the interactions of water molecules following the laws of physics and chemistry. No two are exactly alike, yet each is a masterpiece of natural geometry. They exist for only a brief time before melting, ephemeral beauty that appears and vanishes according to the whims of weather. When you catch a snowflake on your glove and examine it before it melts, you're looking at a structure whose symmetry is determined by the quantum mechanical properties of how hydrogen and oxygen atoms bond, whose shape is determined by the thermodynamics of phase transitions, and whose exact form is determined by the chaotic, unpredictable journey that particular snowflake took through the atmosphere. You're holding, for just a moment, a tiny intersection of quantum mechanics, thermodynamics, chaos, theory, and crystallography. All of that complexity, all of that science, condensed into something you can see with your naked eye, something you can hold in the palm of your hand. The six-fold symmetry of snowflakes also appears in other ice crystals. Frost forming on a window often shows hexagonal patterns. Ice crystals in cirrus clouds are hexagonal plates that catch the sunlight and create phenomena like sundogs and halos around the sun or moon. Even the ice in your freezer, if it forms slowly enough, will show hexagonal crystal structure, though you'd need a microscope to see it clearly. This is because all ice, at atmospheric pressures, has the same hexagonal crystal structure. There are other forms of ice, called ice 2, ice 3, and so on up to ice 15, that form under extreme pressures, or at extremely low temperatures, and have different crystal structures. But the ice we encounter in everyday life, the ice in snowflakes and glaciers and your iced coffee, is always hexagonal at the molecular level. The symmetry of snowflakes reminds us that beauty and science aren't separate. The same principles that physicists use to understand crystal structures and molecular bonding are also the principles that create objects of breathtaking beauty. Nature doesn't make a distinction between what's useful and what's beautiful. It simply follows the laws of physics, and sometimes, often really, those laws produce patterns that captivate human minds and inspire human hearts. Breaking symmetry, the power of asymmetry. We've spent considerable time exploring symmetry in all its forms, marveling at the balance and order it represents. But now, let's examine the opposite, asymmetry, the deliberate breaking of symmetry, and discover how this too can be a powerful adaptation. In the animal kingdom, asymmetry is rare. As we've discussed, most animals are either bilaterally or radially symmetrical. But there are notable exceptions, organisms that have either never developed symmetry or have lost it over evolutionary time. And, in some cases, asymmetry provides unique advantages that symmetry cannot. The most asymmetrical animals are the sponges, members of the phylum Porifera. Sponges are ancient creatures that have existed for over 600 million years. They're among the simplest multicellular animals, lacking true tissues, organs, and nervous systems. A sponge is essentially a collection of cells working loosely together, organized around a system of water channels. Sponges have no consistent body plan. Each individual grows into a shape determined largely by its environment. If a sponge larva settles in a crevice, it grows to fill that crevice. If it settles on an open rock surface, it might grow into a rounded mound or a flattened encrusting form. Some sponges grow upward into tubes or vases, while others spread laterally. There's no internal blueprint dictating a particular symmetry. This asymmetry reflects the sponge's lifestyle. Sponges are sessile, filter feeders. They pump water through their bodies, filtering out food particles and oxygen. They don't move, don't chase prey, don't flee from predators. The advantages of symmetry, like coordinated movement or directional sensing, don't apply to them. Asymmetry allows them to be maximally flexible, adapting their shape to whatever substrate they grow on. Among animals that typically display symmetry, there are also instances of adaptive asymmetry. Flatfish, like flounders and halibut, start life as bilaterally symmetrical larvae. But as they mature, they undergo one of the most dramatic metamorphoses in the animal kingdom. One eye migrates across the top of the skull to join the other eye on the opposite side. The fish then tips over and spends the rest of its life lying on one side on the seafloor. This asymmetry is an adaptation to a bottom-dwelling lifestyle. By lying flat against the sediment, the flatfish can hide from predators and ambush prey. Having both eyes on the upper side allows the fish to see what's happening above it while remaining camouflaged against the bottom. The lower side, pressed against the sediment, loses its pigmentation and becomes pale. The upper side develops coloration that matches the seafloor. Another example of adaptive asymmetry is found in fiddler crabs. Male fiddler crabs have one claw that's enormously enlarged, sometimes as large as the rest of the crab's body. The other claw remains small and is used for feeding. The large claw serves two purposes. It's used in combat with other males, and it's used in courtship displays to attract females. The asymmetry is striking and functional. Having one massive claw is more effective for display than having two medium-sized claws, because it creates a more dramatic visual signal. Interestingly, which claw grows large is random. Some male fiddler crabs have an enlarged right claw, others have an enlarged left claw. If a male loses its large claw, the small claw will grow to replace it, becoming the new large claw. This demonstrates that the asymmetry isn't genetically hardwired to be left or right. Instead, there's a developmental program that says, make one claw large, and the specifics of which claw are determined by chance or by early growth conditions. Crossbills, a type of finch, provide another striking example of functional asymmetry. These birds have beaks where the upper and lower mandibles cross each other, hence the name. The direction of crossing, whether the upper mandible crosses to the left or to the right, varies among individuals. This asymmetric beak is a specialized tool for extracting seeds from conifer cones. The crossbill inserts its closed beak between the scales of a cone, then opens the beak, using the crossed mandibles like a lever to pry the scales apart and expose the seeds inside. Even in humans, we see asymmetry that's functionally important. Our internal organs are arranged asymmetrically. The heart is positioned toward the left side of the chest. The liver is primarily on the right side, while the stomach is primarily on the left. The left lung has 2 lobes, while the right lung has 3. This asymmetric arrangement is highly consistent across individuals, suggesting it's adaptively significant. The asymmetric placement of organs in humans and other vertebrates is established very early in development through a complex cascade of genes and signaling molecules. If this process is disrupted, individuals can be born with situs inversus, a condition where the organs are mirror reversed from the normal arrangement. Interestingly, people with complete situs inversus, where all organs are reversed, usually have no health problems. This suggests that it's not the specific left-right arrangement that matters, but rather having a consistent asymmetry. The brain shows asymmetry too. In most people, the left hemisphere is dominant for language processing, while the right hemisphere is more involved in spatial reasoning and pattern recognition. This lateralization of brain function allows for specialization. By having different hemispheres handle different tasks, the brain can process information more efficiently. Asymmetry even appears at the molecular level. Many biological molecules exist in 2 mirror image forms called enantiomers. For instance, amino acids can be either left-handed or right-handed. But in living organisms on Earth, almost all amino acids are left-handed. Sugars, conversely, are almost all right-handed. This molecular asymmetry is fundamental to life. Proteins built from right-handed amino acids would fold differently and wouldn't function properly. DNA built from left-handed sugars wouldn't form the proper double helix. Why living organisms settled on one form rather than the other is one of biology's enduring mysteries. It may have been random chance early in life's history. Once one form became dominant, switching would be nearly impossible because all the existing biochemistry depends on that particular form. This is an example of historical contingency in evolution, a choice that might have been arbitrary at first, becoming locked in and forming the foundation for everything that came after. In evolutionary terms, asymmetry can be thought of as a special case of symmetry breaking. Evolution generally favors symmetry because it's often efficient and functional. But in specific circumstances, breaking that symmetry provides advantages. A flatfish gains camouflage and predatory effectiveness by becoming asymmetric. A fiddler crab gains a more impressive courtship display by making one claw huge. A crossbill gains access to a food source that other birds can't exploit by developing a crossed beak. These examples show that evolution is pragmatic. It doesn't follow rules for the sake of rules. If breaking symmetry provides an advantage, natural selection will favor asymmetry. The prevalence of symmetry in nature isn't because symmetry is inherently superior. It's because, in most circumstances, symmetry happens to work well. But when it doesn't, Evolution finds other solutions, including asymmetric ones. This flexibility is part of what makes evolution so powerful as an explanatory framework. Evolution doesn't aim for perfection or elegance. It aims for whatever works in the moment, given the constraints of physics, chemistry, and developmental biology. Sometimes that means perfect bilateral symmetry. Sometimes it means radial symmetry. And sometimes it means throwing symmetry out the window entirely and building something asymmetric, irregular, and perfectly adapted to a specific niche. As we observe nature, it's worth remembering that every pattern we see, symmetrical or not, is the result of millions of years of trial and error, of organisms competing and reproducing, of environments changing and populations adapting. The patterns that persist are the ones that work, and working means many different things in different contexts. For a sponge, having no symmetry works. For a butterfly, having bilateral symmetry works. For a snowflake, having sixfold radial symmetry works. There is no single answer, no universal truth. There is only what functions in a particular place, at a particular time, for a particular organism. The Symphony of Symmetry. As our journey through the world of symmetry draws to a close, let's step back and consider what we've discovered. We've explored bilateral symmetry in animals, seeing how it enables directed movement and the evolution of complex brains. We've examined radial symmetry in organisms that interact with the world from all sides equally. We've uncovered the hidden bilateral organization within the seemingly radial echinoderms. We've seen how flower symmetry influences pollination and plant reproduction. We've followed the Fibonacci sequence and fractal patterns as they appear in everything from pine cones to galaxies. We've traced the hexagonal symmetry of snowflakes back to the molecular structure of water. And we've discovered that asymmetry, too, has its place and purpose in nature's design. What emerges from this exploration is a picture of nature as an improviser, working with the materials and constraints available, finding patterns that work and repeating them across scales and contexts. Symmetry isn't imposed on nature from above by some external designer. It emerges from within, from the properties of matter and energy, from the logic of self-organization, from the ruthless editing of natural selection over countless generations. The power of symmetry lies in its efficiency. A bilaterally symmetrical body plan allows an animal to move forward with coordinated power, balancing thrust on both sides. A radially symmetrical body plan allows a sessile organism to defend and feed from all directions without wasting energy on unnecessary structures. The hexagonal symmetry of ice crystals is the lowest energy configuration for water molecules at freezing temperatures. The Fibonacci spiral of a nautilus shell allows continuous growth without requiring remodeling of earlier structures. But symmetry is more than just functional. It's also beautiful. Humans across all cultures respond to symmetry with pleasure. We find symmetrical faces more attractive. We incorporate symmetrical patterns into our art and architecture. We're drawn to the spiral of a galaxy, the radial symmetry of a flower, the intricate branches of a snowflake. This aesthetic response isn't arbitrary. It's rooted in our evolution. Our brains evolved in environments filled with natural patterns, including symmetrical ones. Our visual systems are tuned to detect these patterns quickly and efficiently. Symmetry signals health. A symmetrical organism is likely to be well developed and free of disease or injury. Symmetry signals order in a complex world. It helps us make sense of what we're seeing by revealing underlying structure. Some researchers have suggested that our appreciation for symmetry is actually a byproduct of how our brains process visual information. Symmetrical images are easier to process and remember than asymmetrical ones because we only need to encode half the information and then mirror it. This cognitive efficiency might translate into a feeling of pleasure. What feels good is what's easy for our brains to handle. But beyond aesthetics and efficiency, symmetry reveals something deeper: the orderliness of the universe itself. The fact that similar patterns appear at vastly different scales, from molecules to galaxies, from snowflakes to spiral nebulae, hints at underlying principles that unite all of nature. The mathematics that describes a Fibonacci spiral in a shell is the same mathematics that describes the spiral arms of a galaxy. The crystallography that explains snowflake symmetry also explains the structure of minerals and metals. This universality suggests that we live in a universe with deep regularities, with laws that apply consistently everywhere and at all times. Symmetry is one way these laws manifest. When we study symmetry in biology, we're not just learning about living things. We're learning about physics and chemistry, about how matter and energy organize themselves, about the constraints and possibilities that shape everything that exists. The interplay between symmetry and complexity is particularly fascinating. Simple symmetrical patterns, repeated and elaborated through processes like fractal iteration, can generate structures of incredible complexity. A tree's branching pattern starts with a simple rule. When a growing tip reaches a certain size, it splits into two. Apply this rule thousands of times, and you get the complex architecture of a mature tree with thousands of branches. The pattern is both simple and infinitely complex, both ordered and chaotic. This balance between order and disorder is characteristic of life itself. Living organisms are highly ordered systems, yet they exist in a state of constant flux, constantly consuming energy and matter, growing, moving, changing. Symmetry provides the underlying structure, the framework within which this dynamic activity takes place. A bilaterally symmetrical animal maintains its basic symmetry throughout its life, even as its cells die and are replaced, even as it grows or ages, even as it moves and hunts and sleeps. In many ways, symmetry is the physical manifestation of information. A symmetrical object contains redundancy. The right side carries much the same information as the left side. This redundancy is useful. It provides backup systems if something goes wrong. It allows for balanced function. But it also means that not every part of the organism needs to be specified independently in the genetic code. The developmental program can specify a pattern once and then repeat it, saving genetic information. This efficiency extends to evolution as well. Once a symmetrical body plan has evolved, variations on that plan can be generated relatively easily by modifying how the symmetry is expressed. Changing the length of a leg affects both legs because the developmental program that builds legs is applied symmetrically. This makes it easier for evolution to explore new body forms without starting from scratch each time. As we contemplate these ideas, we're reminded of just how interconnected everything is. The symmetry of your body is related to the symmetry of a fish, which is related to the symmetry of a flatworm, all sharing ancient developmental programs. The spiral of a snail shell is related to the spiral of a fern frond. both expressing the mathematics of the golden ratio. The hexagonal symmetry of a snowflake is related to the hexagonal packing of cells in a honeycomb, both solutions to geometric optimization problems. Nature speaks in patterns, and symmetry is one of its most eloquent vocabularies. By learning to read these patterns, we gain insight into how the natural world works, how it came to be the way it is, and how it might change in the future. We see connections between things that appear unrelated. We discover that the same principles that shape the microscopic also shape the cosmic. This knowledge enriches our experience of the world. When you look at a butterfly now, you see not just a pretty insect, but a living demonstration of bilateral symmetry and its advantages for flight and coordinated movement. When you examine a flower, you see not just colourful petals, but a structure whose symmetry encodes a strategy for reproduction. When you catch a snowflake, you see not just ice, but the crystalline expression of quantum mechanical bonding between water molecules. Every organism you encounter, every natural structure you observe, becomes more than just itself. It becomes a window into deeper principles, a representative of a larger category, an example of how matter and energy can organize themselves into the stunning diversity of forms we see around us. And, perhaps most importantly, studying symmetry reminds us of our own place in nature. We are not separate from these patterns. We are part of them. Your bilaterally symmetrical body developed following the same basic developmental programs as countless other animals. Your aesthetic appreciation for symmetry is rooted in the same evolutionary processes that shape all behavior. Your very existence depends on the molecular asymmetry of amino acids and sugars, on the efficient packing made possible by Fibonacci patterns in your DNA. We are patterns made of patterns, structures built from structures, symmetries nested within symmetries. And by understanding the patterns, we come to understand ourselves. We see that we are not isolated, not exceptional, but deeply connected to everything else that lives, has lived, or will live. We're all playing variations on the same fundamental themes, all shaped by the same physical laws, all participating in the grand improvisation that is life on Earth. As this exploration ends, I hope you'll carry these insights with you. Notice the symmetries around you. See the patterns in trees and flowers, in animals and crystals, in the structure of buildings and the layout of cities. Ask yourself why these patterns appear, what advantages they provide, what constraints they represent. Let your curiosity be sparked by every snowflake, every butterfly, every starfish you encounter. And if you found value in this journey, in these quiet revelations about how symmetry shapes the living world, consider sharing it with others. Like the seeds of a dandelion, spiraling outward in their Fibonacci arrangement, let these ideas spread. Leave a comment with your thoughts, your questions, your own observations of symmetry in nature. Subscribe to Bedtime Biology if you'd like more explorations like this one. And most of all, keep wondering, keep questioning, keep looking at the world with eyes that see the deeper patterns beneath the surface. The power of symmetry isn't just in what it does for organisms, it's in what it reveals to us about the universe. It shows us that order and beauty are woven into the fabric of reality, that mathematics isn't just a human invention, but a language the universe was speaking long before humans existed to hear it. And it reminds us that in a cosmos so vast and complex, we can still find understanding, can still discover connections, can still experience the quiet joy of comprehension. So the next time you see a butterfly, pause, look at the mirror image pattern of its wings, and think about 500 million years of bilateral evolution. The next time you see a daisy, count its petals and watch for Fibonacci numbers. The next time it snows, catch a flake and marvel at its hexagonal perfection before it melts away. These moments of attention, these pauses to appreciate nature's patterns, are themselves a kind of meditation. They quiet the busy mind and connect us to something larger, older, and more beautiful than our individual concerns. Thank you for joining me tonight on this journey through the power of symmetry in nature. May your dreams be filled with spirals and mirrors, with order and complexity, with the endless creativity of the natural world expressing itself through pattern and form. Until next time, keep exploring, keep wondering, and sleep well, knowing that the universe itself is a work of art, painted in the language of symmetry, and forever inviting us to read its elegant script.